Excellent. So Taylor Sundby will be giving our first presentation for the research this morning. Taylor, please.
All right. Well, I appreciate the opportunity for presenting my work, and it's going to be a tough act to follow Will and Eileen, but thank you so much, Will, for sharing that story. It's the inspiration for why we do all of the work that we do. So my name's Taylor Sundby. I'm one of the fellows working in Jack Stern's lab at the NCI. And today I'm going to talk about the use of something called cell-free DNA to distinguish cancer, called MPNST, from its benign precursor lesion, or plexiform neurofibroma. And this is all work that was made possible through the generous funding of the CCF NextGen grant. So thank you again for making this work possible. So I have nothing to disclose. So NF1, or neurofibromatosis type 1, was introduced in our first talk, but it is actually the most common genetic predisposition syndrome. And what I mean by that is these are individuals who have a mutation in their germline DNA, so it's in all of their cells, that alters cell signaling and increases the risk of cancer later in life. In this particular case, in NF1, it is a gene mutation in NF1 that causes it. There's a number of comorbidities associated with this, but the ones that we're going to focus on today are something called plexiform neurofibromas and MPNSTs. So plexiform neurofibromas occur in about 50% of patients with NF1. They're benign growths, so they're going to stay in place, they're not going to invade, they're not going to metastasize, but they can grow quite large and cause a number of morbidities unto themselves. So Dr. Wiedemann, who's in the audience, has done a lot of work on plexiform neurofibromas. But about 15% of patients with NF1 go on to develop something called a malignant peripheral nerve sheath tumor. So these are aggressive soft tissue sarcomas that have an abysmal 5-year overall survival of right around 20%. It's the #1 cause of mortality in NF1, and to a large extent that's because they're resistant to radiation therapy and resistant to chemotherapy. So really the gold standard treatment is early surgical resection. Early surgical resection, though, of course requires early diagnosis. And that's the trick, because in the setting of NF1, these MPNSTs arise from within these plexiform neurofibromas. So you have two distinct tissue types intermixed, making it very difficult to distinguish them both on imaging and then also on tissue biopsy. So I'm going to pause really quickly before I go into this slide just to introduce the ideas of sensitivity and specificity. So sensitivity is a test or an assay's ability to identify all of the cases in a population. So if you imagine that you have 10 people, 6 of whom have cancer, a highly sensitive test might identify 8 people as having cancer. So yes, it captures all 6, but at the cost of specificity. So 2 people who don't have cancer are called as having cancer. Specificity, on the other hand, is the ability to correctly identify patients as having cancer or not in our case. And so that same population of 10 people with 6 people with cancer, maybe you say 4 people have cancer. And yes, all 4 of those have cancer, but there's 2 that you miss. And so our current standards of therapy, or current standards for diagnosing MPNST, are imaging and biopsy. So PET imaging is something that has high sensitivity, but there's a lot of issues with it because there's a lot of differences in what amount of glucose uptake is considered most likely to be MPNST. That varies even within institutions and machines. And MRIs are sort of middle of the road. They have decent sensitivity and decent specificity. So it'll capture a lot of the MPNSTs, and it will probably tell you whether it's an MPNST or not. Tissue biopsies, on the other hand, are high positive predictive value, which is just another way to say that if it tells you it's MPNST, it's very specific. It's probably an MPNST. But they have relatively low negative predictive value. That's to say that if it says it's a plexiform neurofibroma, it might still be an MPNST. And you can sort of see why by looking at the slide on the right. So you can see this plexiform neurofibroma tissue, the PN, directly abuts the MPNST tissue. So you can imagine that if you were to biopsy, if you're just a few millimeters towards the right-hand side of the slide for us, you're going to get the diagnosis of a plexiform neurofibroma when there is, in fact, an MPNST there. Beyond that, tissue biopsies are invasive. They require anesthesia for a lot of our pediatric patients, and that's a non-trivial fact. And so the molecular pathogenesis, or the genetic changes that accompany transformation from plexiform neurofibromas to malignant MPNSTs is actually pretty well described. So plexiform neurofibromas have second loss of that NF1 gene. Atypical neurofibromas, which are an intermediate lesion that we'll largely skip over in this talk, but hopefully in future symposiums I can talk to you a little bit about have loss of CDKN2A, and then MPNSTs have loss of the PRC2 complex gene, so this controls the epigenetics, P53, and then they have increasing aneuploidy. And the aneuploidy just means that normally you have 2 copies of each gene. You can have copy number gains, so more than 2 copies, copy number loss, less than 2 copies, and aneuploidy is sort of the catch-all phrase for that. And so what we see in tissue from MPNST patients is that going from plexiform neurofibromas to atypicals to MPNSTs, we have increasing number of copy number variation, increasing aneuploidy at an almost exponential rate. So what we hypothesize is that we can see these differences in aneuploidy and determine whether something is MPNST or plexiform neurofibroma. Moreover, we can do this non-invasively by using something called cell-free DNA. So to just briefly introduce the idea of cell-free DNA, these are fragments of DNA that are in circulation. They're in your blood, they're in your CSF, they're in your urine. And they're released from both normal, healthy cells. So in fact, if you go out exercising, you will increase the amount of cell-free DNA in your body, but also from tumor cells. And it captures all of the cells in your body. So it doesn't have that same spatial bias that you have with a tissue biopsy. And importantly, we see that it actually recapitulates the genomic landscape, so the same patterns of methylation, aneuploidy, mutations that you see in those tissues themselves. So in this study, we had a group of healthy volunteers, patients with benign plexiform neurofibromas, and patients with MPNST. From all of these individuals, we took some blood, we separated out the plasma, isolated the DNA from that, and then we took something called the Buffy coat, which is the white blood cells, and that tells us what's going on in the germline DNA, not just in the tumor. And then for the patients that we had it, we also looked at their tissue, whether it's a biopsy or a resection. We isolated DNA from all of these patients, made libraries, and did something called ultra-low pass whole genome sequencing. And so here we sequenced to a depth of 2.2x. And basically what that means is that each letter that makes up a gene is a base pair, and every time you read that, you get 1x. So a lot of times people will read each base 30 times, 30x. We skimmed the genome basically, right? We read each word twice. And I'll talk a little bit more about fragment selection, but from this sequencing, we looked at copy number alteration, so this is the aneuploidy, came up with a score called the tumor fraction score, which is a measure of the overall aneuploidy in a genome. So not just saying this gene has more copies, but overall what's going on in the genome. And then correlated that with imaging and clinical course. So this is an example of one patient. So on the top line, you can see their tumor tissue DNA. And so each of these dotted lines represents one of their chromosomes. So you have all 23 chromosomes going across. And blue is diploid, so that's 2 copies, that's normal. Your red and burgundy are copy number gains, and your greens are copy number losses. And you can see, as expected, there's a lot of aneuploidy in that tumor DNA. Importantly, when we look at that second line, which is their cell-free DNA, we see, again, a lot of aneuploidy, and it's in the same regions, the same chromosomes. So this seems to represent what we see going on in the tissue, which is the whole basis of our assay. So we're excited to see that. And interestingly for this patient, as they received treatment and responded, you can see that the amount of red and green on that plot goes down, so there's less aneuploidy. Their germline DNA is on the bottom. It's all blue, as expected. It's diploid. And then you can see our tumor fraction scores on the side. So yeah, it's higher in the tumor, but like we talked about, everybody has normal cell-free DNA, exercising, everything else increases that. And so we expect there to be a little bit of dilution in your pre-treatment cell-free DNA, and it goes down, and in the genome, in the germline DNA, there's nothing. So this is what we expect. So looking at these tumor fractions from all of our patients and all of our samples, we group those all together, and we create something called a receiver operator curve, or ROC curve. And essentially, this is a way to march up and down to find that cut point, above which you say this is likely an MPNST, below which you say it's likely a plexiform neurofibroma. And you can sort of adjust that cut point to find the specificity or sensitivity that you're after, which we talked about earlier. So when we looked at that initially, we saw that we don't perform that well relative to the imaging and to the biopsies. So we had sensitivity of 69%, fine, acceptable, but specificity of only 50%. So this means a lot of people would end up undergoing surgeries who didn't in fact have MPNST. But what we hypothesized is that this is because we're looking for that proverbial needle in the haystack, right? There's a lot of normal cell-free DNA, and that tumor DNA is hard to find there. And so looking a little bit closer at our data, we saw that all of the fragments of DNA. So remember, circulating DNA or fragments is not the whole genome in one big box, but all of those fragments actually differ a little bit by size. And so in the bottom, so on the x-axis here, we have fragment length. Longer is on the right-hand side, shorter on the left. On the y-axis, we have density. So how often do we see this specific fragment length? And you can see that in our shorter fragment lengths, so towards the left, we have more MPNSTs, that's the red, relative to our plexiform neurofibroma and healthies. Another way to look at that is a log-to ratio mean density curve, and basically this just means that as you march along that x-axis of fragment length, the color that you see is what you're more likely to have at any given fragment length. So red represents MPNST, black plexiform neurofibroma. So using this data, what we said is fine, if we're looking for a needle in a haystack, let's make that haystack a little bit smaller. And so we did what's called in silico size selection. So we only looked at the fragment lengths between 90 base pairs and 150 base pairs, and thereby enriched for MPNST. Using in silico size selection, we see that our sensitivity and specificity goes up. Our specificity is 91%, sensitivity 58%. And I also want to point out that this is real world data. In other words, we're not cherry picking our best value. So these are the pre-treatment data for every person. So if I were to take my highest tumor fraction from each patient, and these patients we captured multiple points on, we actually have a specificity of 97.5% and a sensitivity of 66%. But that's not what it's going to be like in the clinic, so that's not what we focus on here. And importantly, so when using that cut point on that curve, we see that those that are tumor fraction high, so they're above the cut point, we call that MPNST, we see the genomic changes that we expect in tissue MPNSTs. So we see 8q gains, that's in red. We see 9p losses in blue, that's where CDK and 2A and some of the other genes we talked about are. We see gains in chromosome 17, which is a little bit hard to see, but that's that other big red stack on the right. And then we see loss in NF1. So all of these are what we expect. And important to point out here, this is aggregate data, so this isn't just one person who I'm showing you. This is the reads from all of our study. Our tumor fraction low, so classified as plexiform neurofibromas, don't have those 8q gains. They don't have the 9p losses, but they do still have that NF1 loss. So that's also what we expect. So we seem to see the changes that we'd expect in tissue in the plasma. And our healthy donors look nice and flat with normal DNA, which I was happy to see because I'm one of those patients too. And then this is to say that when we look at this data, fantastic. It looks good on its own, but what does it look like when we compare it to clinical course? So I'm going to show you 3 quick vignettes, and I'm happy to talk about more later on. But this is a patient who had a MPNST in their pelvis. They had that resected, and on imaging, there was no evidence of disease. We can see that in the blue line, so that's something called SLD, which is essentially a measure of tumor size on imaging. And then in the red line, that's our tumor fraction. So we can see that early on, so on the far left, both of those are 0. No tumor fraction detected, nothing on imaging. As we march along, we can see that their imaging continues to show no evidence of disease, but their tumor fraction actually goes up. And in fact, we see on the next set of imaging that's done about 3 months later, they do in fact have metastatic disease. So this vignette, it's one patient, but it seems to suggest that we're able to detect metastatic recurrence before potentially imaging can detect it. This is another patient, and what we see importantly here is that tumor fraction isn't a fixed number. It changes with the patient's course. So they underwent 3 different lines of chemotherapy. So during the first 2 lines of chemotherapy, you can see the blue line, which represents their imaging disease, continues to go upwards. They're progressing. Their tumor fraction, likewise, goes upwards with that. And as they respond, thankfully, to the 3rd line of chemotherapy, their imaging goes down and their tumor fraction goes down as well. So you can, I don't know if I have a pointer, but you can essentially see, so red and blue are essentially running in parallel to each other. This 3rd patient is a patient who seemed to be responding or stable to responding on imaging. Again, that blue line is stable, maybe a little bit down. So they had significant disease in their leg and wanted to have a quality of life amputation. So it wasn't necessary for their medical care, but they were going to have an improved quality of life. And because that imaging looked good, the decision was made that we can go ahead and hold chemotherapy and do this palliative surgery. But what we see is that had we been able to look at the tumor fraction in real time, that's actually beginning to go up during this same interval. And sure enough, this patient unfortunately had metastatic progressive disease during that chemotherapy hold, which was evident in their tumor fraction, which we looked at again retrospectively. So we looked at after the fact. This is not yet a clinical test. And this essentially ties all that together to say that, yeah, I picked out 3 cases, but this is true for many of our cases. So on the left, this is a correlation. You can see the imaging, so bigger on the right-hand side of the x-axis, smaller on the left-hand side, and then tumor fraction on the y-axis. And we have a significant correlation between our sum of longest diameters, or imaging size, and tumor fraction. On the right-hand side, so this is classified by RECIST criteria, which is a way that clinicians can essentially say, is someone progressing stable or responding to disease? And you can see that, for instance, I'll just go through the top one, but the smallest dot is 1. That's their lowest tumor fraction, and everything's normalized to that. And in this top one, they have low tumor fraction when they have partial responses, stable disease, and complete remission. But then we see that increase once they have RECIST criteria for progressive disease. And that holds true for all of these patients we highlight here. So in the end, what we see is that cell-free DNA is a way to non-invasively distinguish malignant MPNST cancer from its benign precursor lesion, plexiform neurofibroma, with 91% specificity. We see that there's differences in tumor fraction as well as fragment length between healthy controls, plexiform neurofibromas, and MPNSTs. We see that both our plexiform neurofibromas and MPNSTs have loss of NF1, which we expect, but we don't see that in our healthy. So we can detect clinically significant mutations as well, even at the skimming of the genome. And then looking a little bit closer, we see again that we have the changes that we expect in MPNSTs in our tumor fraction high, but not in our tumor fraction low or healthy controls. Imaging and tumor fraction correlate significantly. And then on case series, we see some evidence that, that this effectively tracks treatment response. It can detect minimal residual disease, and potentially it can perform better than radiographic relapse or radiographic imaging at detecting early relapse. So this is all the start of the project. This is actually currently in review, so hopefully will come out for publication soon. But one thing that obviously we want to continue to work on, and we're expanding on by using methylation changes and other changes that were alluded to in talks earlier, is that sensitivity of 58%. So there's still room for improvement, but we feel confident that we have something that performs as well as current standards and non-invasively and at less cost. So of course this was a big project, so all of this work was done in collaboration with Dr. Herbie and Dr. Chinjari at WashU St. Louis. Some of the samples also came from Dr. Pertilis in the NF1 biospecimen repository at Hopkins. I want to thank everybody in my lab. I work in Jack Stern's lab. Carly, Shuan, Bega, Hyen have all been invaluable at helping me advance this work. Baltran's group does some of our sequencing. Dr. Wiedemann has been on my scientific oversight committee with Paul Meltzer and has truly helped guide me through all of this. And of course, I want to thank CCF and all of the donors who made this work possible. Thank you.
All right, it's open for questions.
Oh, perfect.
Go for it. Oh, yeah.
Okay. Yeah, this is for the people in the room. Use the microphone because the people on the virtual wouldn't hear you otherwise. Okay. So I have 2 questions. The first one is, can you go back when you say tumor fraction?
Yeah.
How are you defining that? Is it with 1 marker? Is it 2 markers? Is it a menu of choices? How do you define tumor fraction? That's the first question.
So it's a great question. So essentially what we do is we use something called hidden Markov modeling to look at how this is relative to our normal controls across the entire genome. So it's essentially looking at each of those bins, which are 1 megabase bins because we're only skimming, and integrating all of those signals to come up with a score. So it represents the entire genome. But what you're alluding to is exactly where we're going with our future work, is we know that there are certain parts of the genome that are going to be much more important. So going in greater depth on those specific parts, which we now have the data to tell us what parts those are, may be a way to improve our assay.
So that's a great lead to the second, which is, have you started to look at this as a predictor for MEK inhibitor responses? Because obviously that's tied to Dr. Wiedemann's work, right?
So this is certainly an idea that we're beginning to introduce, and I am using similar assays for treatment response in other types of cancers. Actually, working with with Gene on one of his neuro-oncology tumors or trials. But yes, we hope to take it that way. We haven't gone there yet, but that's an area of active conversation.
Taylor, I have a quick question for you. So I'm curious why you think that the MPNSTs, when their DNA is in the circulation, exists in that shorter fragment preferentially. What is it about the tumor, and maybe what do you think explains that difference? It's such an important part of improving the performance of your assay. I just wonder why that is the case.
It's true, and it's hard because it's actually something that's been shown in other cancers as well. So it's been reproduced in other cancers. It's not incredibly well understood. So we know that a normal circulating DNA fragment is about 160 base pairs. So it's essentially a nucleosome. So it's a fragment of DNA attached around these two histones, right? But we know that there's differences in nuclease activity, so what chops up the DNA in some tumors, but not all tumors. And the fact that we saw differences between our plexiform neurofibroma and healthy size as well suggests that any, quote, abnormal tissue might have some changes in their nuclease activity that chops it up a little bit differently. But I'd love to tease that apart a little bit more.
Yeah, interesting. Brigitte?
Yeah, I just wanted to... I wanted to take the opportunity to thank the CCF as well. While this is focused on a rare tumor, on MPNST, it's a huge clinical problem, and we literally have seen kids where we know they are at high risk. We do every half-year whole-body MRI, and in between that time period, they develop an MPNST, and at that time, most of the time, they cannot be cured. So having something that's cheaper, that can be done from home and more frequently would help. And I don't know, Taylor, if you want to briefly mention that, I think you did, but that you work on other tumors to use the same, like an assay that would be applicable to other tumors to essentially look at not only diagnosis, but response on therapy, which I think is really cool.
Absolutely. So I think there's a lot of potential for this technology, in part for exactly what you said. So these tubes that we can collect in are stable for 7 days. So they can have it drawn in Bakersfield, California, and send it to us, and we can still do that assay. So that's great. And yeah, so right now we are using this. So currently we have 7 clinical trials where I'm looking at cell-free DNA and how that responds to treatment response, but also for, like Gina was saying at the beginning, we know that these baskets are full of a lot of different cancers, and so trying to tease apart what different type of cancer is this, and is it more likely to respond or not. So I think there's a lot of applicability to clinical trials, and then we're also working at this for surveillance in MIN1, MIN2, MIN2B. There's a lot of different cancer predispositions that I'm trying to create similar assays for.
Go ahead, Kurt.
Yeah.
Thank you. Lovely talk and work,
Thank you.
Taylor. I was just thinking about trying to advance this to looking for what you're looking for is second hits, third hits, et cetera.
Right.
And some of these are defined by relatively discrete mutations, some of them not. So those you could design, simple PCR, multiplex PCRs for to supplement, perhaps confirm the sequencing?
You're exactly right. So there's 2 different approaches that you can use there. So you can use something called droplet PCR, which is what you're alluding to, so that you can go really deep. So instead of reading 2 times, you read every base pair 3,000 times, really. And so that's one option. Another option is we actually have a targeted panel. I didn't show it today, but something called CAPSeq. So there, the beauty of that is you do get some off-target reads as well, and you can actually get a lot of information from that. So you can both generate hypotheses while really focusing on some of the areas that, like you said, are known discrete bad players. Cool.
Awesome. One more quick question for you, if you don't mind. So I was fascinated in the pattern of change in your copy number variation after treatment. And it seemed like after treatment, the copy number gains were more likely to be lost than the copy number losses. It seemed like after treatment, the losses persisted and the gains were gone. Is that? Do you think that's just an artifact of that particular case, or that was several cases?
It was seen in several cases, and I think that's an area to investigate further. Honestly, I'm sure that I've seen that pattern before, but thank you for introducing that. It's something I want to look at more.
Well, it suggests that maybe there's clonal heterogeneity, and that there may be tumor cells that are driven that have that the tumor cells that have clonal gains may be more sensitive to therapy than those with clonal losses. And it might be worth doing some more deep sequencing, some single cell work to try to sort that.
And we know we can track clonal changes throughout, so I think you're exactly right. That's probably what we'll look at. Yeah.
Great. Any other questions? I don't see any from the virtual audience. Just to remind the virtual audience, if you can hear me, please submit your questions as well so we can get them in. Taylor, great job.
Thank you, guys.